Nong's herbal classic. Apart from Ganoderma lucidum, he also documented 300 types of medicinal herbs and plants in that book, and the translated version is still available today. This is Emperor Shi Huang of the Qing dynasty who built the Great Wall of China. So he was the first emperor or the royal member who consumed the decoction of Ganoderma lucidum. So these are the two statues at the Alfei Biotech Park in Nantong, China, showing Emperor Shi Huang and Emperor Yan holding the precious medicinal mushroom. At that time, only the royal members can consume the decoctions or the preparation of Ganoderma lucidum for eternal life and longevity. However, anyone can consume that today after 2000 years. So this is the largest mushroom farm in Malaysia, owned by Gano Farm R&D Sdn Bhd, located in Tanjung Sepat, Selangor. The size of this mushroom farm is about 30 football fields, 30, run by 80 employees. So these are the mushroom houses. The houses are kept warm, moist, and dark throughout the cultivation process. These are the baby mushrooms of the Ganoderma lucidum. The houses are built in the oil palm plantation. So the reason is to get the agricultural waste residues as the substrate for mushroom cultivations. So I took some of the medical students for the elective project 8 years ago to conduct some of the studies in the mushroom farm. These are the substrates. So we use the agricultural waste materials. The major components comes from the rubber wood sawdust from the rubber industry that make up about 85 to 90% of the composition, followed by 5 to 10% of the oil palm frond parenchyma tissue, the OPFPT. So all these crops, after getting the yields, we can recycle the materials such as the OPFPT and empty fruit bunches, as well as the rubber wood from the rubber industry. A small percentage of the rice bran, in Bahasa, we call it dedak padi. So after the ingredients are mixed well, we perform the bagging and sterilization in a huge steam sterilizer for 8 hours, cooling down, followed by inoculating the mushroom bag with the mycelial spawn. The word mycelium means the vegetative state of the mushroom before forming the fruit bodies or the mushroom that we see today. Okay, this is the baby mushroom or the primordial state of the Hericium erinaceus. The mature fruit bodies, yellowish or brownish in color. It is also known as the lion's mane mushroom among the Westerners because the spines are longer compared to the one cultivated in Malaysia. In Malaysia, we call it cendawan bunga kobis, monkey's head mushroom. So these are the usual name for Hericium erinaceus. In the late 90s to early 2000s, my study focuses on the optimization of the mycelial growth in liquid submerged fermentation as well as the solid submerged fermentation. So there's nothing much about the medicinal properties of it. Our aim was to upscale the production of the mycelium, testing the composition of the liquid medium, which is suitable for the growth of this mycelium. So with this liquid submerged fermentation, we can recover the useful bioactive metabolites as well as the polysaccharides from the mycelium or from the liquid media itself. So this is the composition. The major one is made up of the dextrose, about 55%, pH 5.5 with 8% initial inoculum. These are the ideal parameters for the liquid submerged fermentation. This is the bioreactor culture. So on the laboratory scale, we have different sizes of the bioreactors. So some of these are available in UiTM Shah Alam, Principal Investigator Dr. Zadariana, 20-liter size, 60-liter size. So the bioreactor system, they are used not only to grow the mycelium or to propagate the mycelium, but also to treat the industrial wastewater. And to conduct these experiments, we need to monitor the pH, temperature, agitation, aeration, substrate utilization, foam formation, and the final product formation. So that is quite laborious. On the other hand, the solid substrate fermentation is useful for the recovery of the lignocellulolytic enzymes. So the powerful side of this enzyme is it is able to degrade the toxic pollutants in the wastewater, the xenobiotics, the polyaromatic hydrocarbons. In the lab scale, we use the oil palm frond parenchyma tissue from the oil palm plantation, inoculated with the Hericium erinaceus mycelium, let it grow for about 2 weeks. The mycelia feed on these waste residues to get the nutrition and the metabolic support. At the same time, the mycelia secrete these enzymes: cellulase, hemicellulase, manganese peroxidase, lactase, and lignin peroxidase. So these enzymes can be used to treat the organic pollutants. These are the agar plates incorporated with the synthetic dyes that belong to the polymeric group of dye. It is usually found in the wastewater generated by the textile industry. Apart from that, the petroleum products, color printing, paper photography, all these also release toxic materials, okay, to our environment. So we found that the mycelium of this mushroom, the Hericium erinaceus, is able to degrade the synthetic dyes shown by the presence of clear zone in the agar plate. So these enzymes actually clear all this toxic waste. So that is one of the application of industrial biotechnology using mushroom. So growing mushroom is not just obtaining the fresh mushroom for sales. It is a permaculture approach. So the remaining residues after growing the mushroom can be recycled as the animal feed, fish feed, incorporated into the animal dungs as the soil fertilizer or the soil conditioner. Once the crops are harvested, we can use the wood residues as the substrate to cultivate the mushrooms. So this is called permaculture with a mycological twist. Mushroom growing is an income generation activity. These are the case studies of growing mushrooms in rural and sub-rural areas to alleviate poverty. So I will take you through my master's project, antimicrobial, antioxidant, and stimulation of neural outgrowth activities of Hericium erinaceus, supervised by Prof. Dr. Vicky Neswari and Prof. Norida Abdullah. In the mid-2000s, we shifted our focus from bioremediation technology to the medicinal aspects of these mushrooms as people are getting more health conscious. So I graduated in 2007. In my study, I compared the different processing methods of this mushroom, oven drying, freeze drying, with the fresh mushroom and the mycelial biomass. In terms of antioxidant activities, we found that the oven-dried mushroom had high antioxidant capability due to the generation of Maillart's reaction products, MRPs. So these MRPs are usually generated during high heat processing, responsible for the flavor, texture, browning of the food, as well as giving high antioxidant activity to the food itself. As for the antimicrobial activity, we found that the freeze-dried mushroom give the best activity against 13 species of bacteria and 3 species of pathogenic fungi. Other than that, we also look at the neuratogenesis activity or the stimulation of neural outgrowth activity in neuronal cell line known as NG1015. And we found that the fresh mushroom were able to stimulate neuritogenesis and also the formation of neurite beading along the neurites. So we proposed the potential use of Hericium arenaceus in enhancing the nerve regeneration process. So that was part of my PhD project supervised by Professor Dr. Vicky Neswari and Professor Dr. Morali. I graduated in 2011. Okay, let me take you through my PhD project. So I used this mushroom to treat the nerve injury following axonomesis. Axonomesis is a type of crush injury. So peripheral neuropathy can be caused by traumatic injury, diabetes, alcohol abuse, chronic kidney disease, and certain medications. And these are the clinical manifestations, including numbing, loss of sensation, impaired vibration. So in my study, I pre-treated the rats for 14 days with the Hericium arenaceus water extract. We actually prepared the polysaccharide from the water extract. Okay. The crush injury was done after that. This is followed by 20 days of oral feeding of the polysaccharide to the rats. So these are the groupings. Hericium group, the positive control group, or the mecobalamin, vitamin B12, and the negative control group without treatment. To prepare the Hericium arenaceous polysaccharides, so these are the steps involved. It started with boiling of the mushroom or the fresh fruit bodies in distilled water for a long duration, about 8 hours. Centrifuge, precipitation, washing, freeze drying. This is followed by dialysis against distilled water in a tubing cellulose membrane for 4 hours to remove the low molecular weight compounds. So we don't need that because our aim is to recover the polysaccharide from this preparation. So the last step was freeze drying of the aqueous solution, okay, to get the crude polysaccharide. So it is a brownish fluffy powder like this, Hericium arenaceous polysaccharide. Okay, how about the procedure for nerve crush? We did the gluteal muscle splitting incision. We lifted up the sciatic nerve. So all these procedures were guided by Prof. Murali, my PhD supervisor. And we performed a crush injury on the common peroneal nerve, about 10 mm from the bifurcation point of the sciatic nerve. And the complete crush was confirmed by the presence of translucent band across the nerve, as well as the dragging of the affected hind limb. As for the motor function, we observed the toe spreading reflex and also walking track analysis. We'll put them on the walking track. So we found that the polysaccharide or the positive control group gave earlier recovery of motor function. As for the sensory function, we placed the rats on the hot plate heated to 50 degrees Celsius. We measured the withdrawal reflex latency. And we found that a better recovery in the mushroom group as early as 8 days, okay, compared to the other groups in terms of the sensory function. So these are the challenges that we faced during the behavioral tests. As for the morphological analysis, we harvested the extensor digitorum longus muscle. So all these are the anterior compartment muscles of the thigh supplied by the deep branch of the common peroneal nerve. In fact, we can also get the tibialis anterior muscles. However, in my study, I employed the EDL muscles. So we harvested the muscles, fixed them in the buffered formal calcium for 6 hours, immersed them in the 10% sucrose. So these protocols are according to Namba et al., 1967. We did the combined silver cholinesterase method to observe the neuromuscular junction. So in this preparation, the end plates are stained brown, the axons are stained black, skeletal muscles are stained yellow. In the normal rats, without injury, without any treatment, so this is how the silver choline esterase stain on the frozen sections of EDR muscles look like. The end plate in brown. So the faded background, yellowish background are the skeletal muscles. These are the axon bundles. Okay, in this study, for the mushroom group, we observe loose bundles of axons, establishing connection with the motor end plate. In contrast to the polysaccharide group, for the negative control group, we observe the Wallerian degeneration, which was still in progress. It involves removing of the myelin debris, okay, by the Schwann cells and the macrophage. So the polysaccharide group or the treated group recovers better, or we can say that enhanced muscle reinnervation and formation of neuromuscular junction by the mushroom group. This is a positive control group, the mecobalamin. We also harvested the lumbar 5 dorsal root ganglia for immunofluorescence analysis. We use these biomarkers, AKT, MAPK, c-Jun, and the c-Fos. So we observe increased expression of phosphorylated AKT in the DRG following crush injury in the mushroom group. In general, the sensory neurons in the DRG can be classified into 3 groups, the small, medium, and the large sizes. The small neurons are responsive against pain sensation or the nociception, chemical and mechanical stimulation. In contrast, the large neurons are responsive against the touch and the proprioception. So we could see that all these biomarkers that are proposed are confined to the small neurons, okay, highly expressed in the polysaccharide group. AKT is responsible for neuronal survival. As for the MAPK, we also observe high signaling in the treated groups, the mecobalamin and the polysaccharide. So MAPK is responsible for neuratogenesis, okay, formation of axons. And these are the markers for immediate early gene response, the c-Jun, also highly expressed in the treated groups, the c-Fos. We also studied the pattern of blood-nerve barrier after crush injury, and we found early restoration of blood-nerve barrier in the treated groups, indicated by the signaling of RECA1. RECA stands for red endothelial cell antigen 1, which targeted on the vascular endothelium. Okay, other than that, we also observe increased expression of nuclear ribonucleoprotein in the regenerating axons distal to the crush injury. Okay, it is enhanced in the polysaccharide group. So the protein synthetic machinery are actually found in the regenerating axons. They are composed of the Golgi apparatus, ribosomes, and the rough endoplasmic reticulum to make sufficient proteins such as cytoskeletal proteins and neurotransmitters during the regeneration process. So the key findings of this study involves the binding of the neurotrophic factors to TRKB receptor, initiating the signaling cascade of RAS, RAF, MEK1 and 2, ERK1 and 2, and translocation of ERK1 and 2 to the nucleus to trigger the immediate early gene response, the c-Jun and c-Fos. Apart from that, the PI3K AKT signaling pathway was also involved to promote neuronal survival. So all these molecular events contribute to enhanced muscle reinnervation, formation of neuromuscular junction, recovery of motor and sensory function. So these are the related publications from this study. A book chapter published by the National Academy of Sciences of Ukraine. One of the images was selected by the IBD calendar. And some of the related projects are conducted by Professor Dr. Amin Abdullah of Biomedical Sciences Department. It was also part of my PhD project. By testing the Hericium erinaceus aqueous extract on ethanol-induced gastric ulcers and wound healing in animal models. Subchronic toxicological assessment of Hericium erinaceus aqueous extract. This project was led by Associate Professor Dr. Rosie Pamela, conducted by Dr. Hari Prasad and Dr. Jagadesh. So in 2016, we used a product known as Nerve Grow. Nerve Grow is a standardized water extract of this mushroom. Actually, I'm no longer using the fresh mushroom for my study, but I focus on Nerve Grow. We initiated 2 agreements with the CEO, Gano Farm R&D Sandra Berhad, Madam Cheng Poguat. We planned the experiments with proper budget, schedule on Memorandum of Understanding and Memorandum of Agreement established in 2018 and 2020 respectively. So this project, okay, the first project using Nerve Grow is this. Hericium erinaceus potentially rescues behavioral motor deficits through ERK CREB PSD95 neuroprotective mechanism in rat model of 3AP-induced cerebellar ataxia. So that is a long title. So this project was led by Dr. Lim Li Wei of Hong Kong U and conducted by his MPhil student then, Ms. Chong Pik Shan. She's now a PhD student. Just a brief about the project. So in this study, we induced cerebellar ataxia, mimicking the acquired cerebellar ataxia by using a neurotoxin named 3-acetylpyridine 3AP. So 3AP damages the inferior olivary nucleus of the medulla oblongata. As we may have already known, this structure of the brainstem sends the climbing fibers to provide excitation to the Purkinje cells in the cerebellar cortex. And the axons of the Purkinje cells are the only output of the cerebellum directed towards the deep cerebellar nuclei in the white matter. And the principal destination of the efferent fibers are towards the thalamus and other regions of the brainstem, reticular nucleus, vestibular nucleus, and the red nucleus. So by damaging the inferior olivary nucleus through the administration of 3AP, we observe abnormal morphology of Purkinje cells and poor motor performance due to a deficiency in cerebellar firing. So this is how we induce the cerebellar ataxia. We perform intraperitoneal injection to the rats following 3AP administration. So IP injection of Nerve Grow can induce this signaling cascade, SOS, RS, RF, MEK1 and 2, ERK1 and 2, and subsequent translocation of ERK1 and 2 to the nucleus to upregulate the neuroplasticity and neurogenesis related genes, namely the TRKB, CREB, nestin, and DCX, as well as to elevate the neuroplasticity related proteins in the cerebellum, ERK, CREB, PSD95. Therefore, in this study, we found a potential rescue of locomotor abnormalities and recovery of motor coordination and balance following intraperitoneal injection of this mushroom. So nerve growth is high in exogenous antioxidants that can upregulate the endogenous antioxidants in the cells. So the findings have been published in scientific reports 2 years ago. We also have 2 in vitro projects related to nerve growth conducted by Liu Ziyuan. Antioxidant-mediated protective role of Hericium erinaceus against oxidative damage in fibroblasts from FRDA patient. A brief intro about the FRDA. So FRDA is a genetic ataxia. It has no cure. So the current medication aim at relieving the progressive symptoms of FRDA patients, okay, including scoliosis, loss of reflex, ocular abnormalities, sexual dysfunction, cognitive disability, and ataxia. So this FRDA is usually diagnosed in the early childhood, and within 10 to 15 years, the patients will be usually wheelchair bound. And the main reason of mortality is cardiac dysfunction. So in my study, we use FRDA patient fibroblasts obtained from Cornell Institute for Medical Research in the United States. We use LBSO, L-butyonyl sulfoxamide, to mimic the actual pathogenesis of FRDA by reducing the level of glutathione and endogenous antioxidants. So the pathogenesis of FRDA started with the mutation in the frataxin gene caused by abnormal GAA trinucleotide repeat in the first intron of frataxin. So this gene encodes mitochondrial protein known as the frataxin, which is important in the formation of iron-sulfur cluster for proper mitochondrial biogenesis and mitochondrial function. However, upon administration of LBSO, we observe a decrease in GSH, depleted endogenous antioxidant activity, increased reactive oxygen species. So these are the damaging effects of oxidative stress caused by LBSO, okay, mimicking the FRDA pathogenesis. And finally, apoptosis, okay, induced by LBSO. So we observed that following administration of nerve growth, we found attenuation of RS production, restoration of mitochondrial biogenesis and mitochondrial function, increase endogenous antioxidant enzymes, namely the SOD and the GPX, glutathione peroxidase. We also found increased expression of nuclear transcription factor, the NRF2 and NRF1, as well as the mitochondrial transcription factor, the TFAM. So TFAM is responsible for mitochondrial biogenesis. So this study is still ongoing. We have not actually completed the whole study. There are many areas yet to be explored for the genetic ataxia. And the preliminary findings was published 2 years ago in Food Science and Technology. So we established this material transfer agreement with Cornell Institute for Medical Research to obtain the FRDA fibroblasts. Finally, this is the project related to nerve growth, neuroprotective effects of Hericium erinaceus against high-dose corticosteroid-induced oxidative stress in epithelial cells. So we have our chemist, our collaborator from the Department of Chemistry, to look into the compounds of nerve growth. So in this study, we use high-dose corticosteroid of 400 micromolar to induce the damaging effects of oxidative stress, mimicking chronic depression. So we've found increased level of ROS, diminished mitochondrial biogenesis and mitochondrial function, depleted endogenous antioxidants, and loss of membrane integrity, okay, that contribute to apoptosis. So this is somehow mimic the HPA axis hyperactivation. So by using this mushroom product, we found an attenuation in the ROS production, restoration of endogenous antioxidant enzyme level, particularly the SOD and catalase, restoration of mitochondrial biogenesis and function, increased level of TFAM, NRF2, and NRF1. So these are the compounds, known compounds actually, nothing is novel here, known compounds, adenosine, heroin 3, heroin 4, and the sphingosine. Somehow, there's no report related to heroin 3 and heroin 4 pertaining to the medicinal properties. So we do invite potential collaborators to look into the medicinal properties of these two compounds, as well as to upscale the production of these two compounds. So the findings that has been published in the BMC journal, and this is the in vivo aspect of the depression led by Dr. Lim Li Wei. So through this mushroom product, we already gathered about, let me count, 8 awards from 2019 to 2022. Okay, 3 awards from this event. One from Malacca International Intellectual Exposition, Malaysia Technology Expo, Scitrax, organized by University Malaysia Pahang, the recent one, MTE, and Inventions Geneva Evaluation Days, a silver award. Other than that, Ms. Chong Pei Shan had also obtained the best MPhil thesis for her work in nerve growth, and Ms. Liu Ziyuan for the poster award in the recently held ISBC. So this mushroom has been featured in the Petri Dish, Research Bulletin, Milestone at Newsletter, Pulse at Rubrica, Hong Kong U press releases. Okay, now I will take you through some of the ongoing projects. Okay, in terms of bone health, we have Dr. Nam Hu Yin as the PI. So in the preliminary study, we observed that Hericium erinaceus enhances osteoblast proliferation and matrix mineralization. Dr. Sharifah Noor from the Faculty of Dentistry is looking at the induction of apoptosis in oral cancer in vivo and in vitro models. As for the zebrafish model of traumatic brain injury, we have Dr. Suzita and Dr. Rozaida. So we invite potential collaborators to work with us on these projects. Other than the hericium, we do have these potential mushrooms, caterpillar fungus, the tiger's milk mushroom, and the Ganoderma lucidum. So we can explore more on that. These are the dishes prepared using the hericium erinaceus mushroom, the herbal soup, rendang. You can pair it with nasi hujan panas and the satay. Okay, I would like to thank Department of Anatomy for the continuous love, guidance, and support. And lastly, I would like to take this opportunity to advertise PhD vacancies at full-time, working on a full-time basis. So if you are interested, please do contact me. Okay, that's all for my presentation. Okay, thank you, MAA.
Thank you very much, Dr. Wong, for your very interesting research throughout past 20-year research journey. So now I would like to invite the participants, attendees for the Q&A sessions. Any questions from the participants, or you can share your thoughts, your opinions with Dr. Wong.
Hi, good morning, Dr. Wong.
Morning, Dr. Siti.
Congratulations and for your outstanding achievement. I think that was indeed a very excellent journey and a lot of outcome and benefit to others too. Okay, I just want to ask, okay, do you have any plan to expand your study in human subjects? And I'm meaning that how do you translate your research into practice, especially for I mean like clinical clinical practice, educational. Maybe you can collaborate with educationists, okay, right, to look at the mental well-being, professional well-being, you know, something like that. Do you have any plan towards that?
Thank you for your question, Dr. Siti. Yes, we do have that plan. Um, okay, for this mushroom, actually, the Gano Farm R&D is working with the TCM practitioner to see whether this mushroom can improve the neurodegenerative disease in a Parkinson's patient and also Alzheimer's disease patient. So they get the positive feedback from the TCM practitioners, but this is without a proper agreement. So from my side, yeah, we wish to conduct that in humans to look at the efficacy of this mushroom, whether that can mitigate the symptoms of neuropsychiatric disorder. So actually, we are applying grants for that. We look into the potential collaborators, probably from the clinicians who are interested. At the moment, we have not conducted any clinical trials. So yeah, we can work together, Dr. Siti. Yeah, thank you.
Thank you, thank you.
Thank you, Dr. Siti, for the question. Any other questions from the floor?
I received a feedback that this is an interesting title because that like indirectly reveal my age. I started this project as my FIP 20 years ago, 2002. So I was 22 years old then, 22 plus 20, I'm 42. So indirectly reveal my actual age. Okay.
So there is one question, Dr. Wong.
Yes, I
Is
saw the
there,
question.
okay, yeah. Is there any
That's fine.
potential toxicity effects of the mushroom been studied so far?
Okay, thank you, Dr. Siti Rosmani. We have conducted the subchronic toxicity test in rat models in 2016. So we did not find any toxicity effects in the in vivo model up to 1,000 milligram per kg rats. Yes. As for the human trials, we do not have that data yet. Yeah. Thank you, Dr. Siti.
Okay, any more questions?
Hello, yes, I have a question. Here is Paxi from UPM, Department of Human Anatomy.
Yes, doctor, you may proceed.
Hi, morning, Dr. Wong. Thank you for sharing the amazing story. It's really great that you have shown the power from how you involve the both in vitro and in vivo model. And may I know, is this part of your work has the potential maybe to apply to the electrophysiology component? That I see that you also collaborate with Prof. Lim Li Wei. I think that he is also mastering in the field of electrophysiology.
Thank you, Prof. Chia. So Dr. Lim is specialized in neuromodulation. We have not looked into the electrophysiology aspect of that. Yeah, probably that will be our future plan. Yeah, we're not sure for now. Yes.
That's exciting.
Okay.
Because myself, we are establishing this lab, and I'm seeing that is there any of the collaboration potential to apply your current component that actually can test on some of our neurological disorder models that's on the mouse model.
Wonderful, wonderful. Yeah, we can talk about that. Yeah,
Oh,
soon.
thank you. Thank you
Yes, yes.
for
Thank you for proposing
bringing.
this idea. Thank you.
All right. So because you're talking about the nerve regeneration, I think that besides all the immunohistochemistry to show the regeneration, maybe more functional output would be the electrophysiology
Yes,
component
yes.
that can enhance the scientific effect of this active compound. Thank you so much. I will stay in touch.
Sure.
Thanks again for sharing such a
Thank you for
beautiful
suggesting that.
story. Congratulations to you and your team.
Many thanks.
Okay, thank you, Dr. Chia. Dr. Wong, there is one more question. Did you
Sure.
check the inflammation in the ataxia model, like microglia number?
We have not done that. The inflammatory pathway. We look into the neuroprotective mechanism and the neurogenesis aspect of this model. So we have not actually looked in the neuroinflammatory pathway of it. Yes. So that is a good suggestion, Prof. Yeah.
Any other questions from the floor?
Hi. Hi, Dr.
Yes.
Dr. Wong. I'm Dr. Pin from
Hi,
the
Pin.
NanoCare. Thanks for the wonderful and excellent talk. It's very interesting. So I have a question. I have two questions here. So I would like to know that since you just now talk about you have tested toxicity on your studies, particular in this mushroom. So do you plan in the future, since you may have a potential to market this product, so do you plan to have to submit or to apply the toxicity level and then studies to the NPRA so that you can like go through the regulatory bodies for the marketing potential purpose?
Yes, Dr. Nam, let me answer your first question. This product has obtained the NPRA approval and KKM
Okay.
approval 3 years ago. So it is a dietary supplement. It is an established dietary supplement registered under the KKM. Yeah.
Oh, okay. So since you are saying that in the future you may have a test in the clinical trial in the human, right? So I think it is a good start to have like go through the regulatory bodies and then later on go to it's easier to the clinical trial later.
Thank you, Dr. Nam, for your suggestion.
My second question is just now, the suggestion from the Prof. Chia also about the electrophysio. So it is a good idea that because my background is about the mechanobiology. So since about the neurodegeneration, maybe it is a good idea or collaboration in using the concept of the mechano stimulation in your studies also.
Yeah, sure. Maybe 3 of us can work together on this project. Yeah. Thank you.
Absolutely.
Thank you.
Absolutely.
It's a very great potential project.
Dr. Nam was my classmate in Form 6 back in 1998 to 1999.
Oh no, indirectly reveal my age also.
Yes, same age.
Thank you, Dr. Wong. Thank you.
Dr. Wong, there is one more question from Dr. Siti Rosmani. May I know how do you convert the effective dose tested in rats into the effective dose in human?
I believe there's a certain calculation for that, but I'm not really familiar. Once I get the potential clinicians to work on these human aspects, I will consult them. At this moment, I really do not have a firm idea on how the calculation work. Yeah. Thank you, Dr. Siti.
Okay, so I think that's all the questions from from our participants today.
Yeah, if you can always drop me an email, yeah, to find out more about the mushrooms or the medicinal properties of that, or if you are interested to have a networking or a discussion, yeah, please do send us an email.
Okay. Thank you, Dr. Wong. Okay, so here I would like to express my heartiest gratitude to Dr. Wong on behalf of Malaysian Anatomical Association for your time and wonderful yet comprehensive remarks on your research. It's a great sharing. We hope that this session has been fruitful and engaging to all our MA participants. So we hope these productive sessions facilitated everyone to be in a happy disposition today. So before leaving, okay, we plan to have photography sessions, okay, as memories. So
Let me start sharing, right?
okay, I request all the participants to turn on the videos to have photo sessions together with our speaker. To see there are a lot of familiar faces here.
Hey everyone, count one. Okay, because this will have one, two, one. Only one. Only first page is video on. The rest is not. So I will count 1, 2, 3. Okay. Mine 1, 2, 3. All right. Then go to the next page. Okay. The next page is actually no video on. Okay, except 1 or 2. Right. Okay, 1, 2, 3. Smile, keep smiling. All right. Okay, that's all. Thank you everyone for your cooperation.
Okay, thank you
This is
very much to
Rani.
all. Okay, before we ending our webinars, to all the MA members, you could also grab this golden opportunity to become our inspirational speaker in our next sessions. So you can, you all can register for become our speaker. The link for the registration for to become the speaker is in the chat box. Okay, we look forward for your active participations in this constructive event. Okay, so with that, ladies and gentlemen, on behalf of MAA, we would like to express our heartfelt appreciations for your contributions and active participation today, especially to Dr. Wong, and have a great rest of your day. See you in the second round of the workshop next month. I am Ms. Rani signing off. Thank you very much to all.
Thank you, Puan Rani. Thank you, Dr.
Thank you.
Norhida. Thank you, MAA. Yes. See you.
Bye bye.
Thank you,
Bye-bye.
Dr. Wong. Bye-bye.
Thank you, Dr. Siti. Bye-bye.
Thank you very much. Bye.